Greetings, friends. Gold here. Just wanted to bring you another little science article. This will be a quick one. New techniques to freeze brain tissue without harm. So the down and dirty on this article is a team of medical researchers at the National Children's Medical Center Children's Hospital at Pudong University in China. They've developed a new compound or a new technique to treat brain tissue that will allow them to be able to freeze this tissue. They used a combination of four chemicals: ethylene glycol, methylcellulose, DMOS, and a chemical called Y27632. They call the solution MEDI. This MEDI solution is used as a bath for the tissue samples, the small tissue samples. They bathe the small tissue samples, or they refer to them as organoids in the article, and they bathe them for a set amount of time. After receiving this chemical bath, they're then able to freeze these small tissue samples. Um, Preliminary tissue freezing was up to 150 days and thawing without damage, and they used it on a live tissue sample, live human tissue sample that is, and they froze it for up to 18 months. Um, so pretty much they developed a technique to preserve small portions of brain tissue. Um, couldn't find any additional information if they've done anything larger. So you're not looking at a full brain preservation here. You're looking at small tissue samples. So the main the main use for this type of treatment or this type of procedure is to collect and preserve different types of cells and tissues for additional research. So they're hoping that they can take different samples, tumors, things like that, or normal healthy tissue, and they'll be able to catalog them and preserve them for longer without having the negative effects of freezing. Because negative effects of freezing of human tissue is once the tissue freezes, the water in the cells will expand, create ice crystals, and puncture the cell walls, causing tissue damage and deformation. Um, with this process, it's blocking that. It's applying pretty much a antifreeze um, chemical to these tissues. The article doesn't state anything about being able to reanimate it, but it just states that it's not damaged in the freezing process. So hopefully this might open up the door to new research on the brain, and maybe we can find out more inner workings of things like that. That's all I've got for today. You take care. Until next time. Goodbye.